Hey there, I'm making this video to explain two important questions that have been asked in board preparatory 1. Double fertilization is the unique feature of angiosperms and the product of this are zygote and primary endosperm nucleus. In the context of this, when hexaploid plant pollinates a tetraploid plant, find out the ploidy of zygote and primary endosperm nucleus through schematic representation starting from ploidy of male and female plant. So the given information is that male plant is hexaploid. Hexaploid, as we know, it is referring to 6N. And the other information given is that the female plant is tetraploid. And also we know that tetraploid means 4N. And we know that the male and female plants will produce gametes upon meiosis. When hexaploid plant undergo meiosis, that is the microspore mother cells undergo meiosis, they produce male gametes. Two male gametes will be produced and the gametes will have the ploidy half of the mother cell, so that is 3N. Both of the male gametes have 3N ploidy, whereas the female will produces gametes upon meiosis. The megaspore mother cells will undergo meiosis to form one female gamete, that is egg. The ploidy will be 2N and two polar nuclei are formed and two polar nuclei will be present in one central cell, so we are writing it as 2N plus 2N. Now during double fertilization, one of the male gamete will move towards the egg cell within the embryo sac and get fused with it, and the process is called syngamy. And the syngamy will result in formation of a zygote. The ploidy of zygote here will be 3N plus 2N, that is 5N. The other male gamete present in the pollen grain will move towards the polar nuclei and get fused with it. Polar nuclei is having 4N and the male gamete is having 3N. So the primary endosperm nucleus, that is the product of triple fusion, is primary endosperm nucleus. Then the ploidy will be 7N. So here we got the answer through the schematic representation. So this is the way you have to write. And the another question is asked is 43rd question. It is asked in two steps, that is categories of host A, B, C into transformants, recombinants and non-recombinants. So here answer is A and B are transformants because both of them are having a plasmid DNA and the host genome. And here the recombinant is only A because recombinant cells are having vector DNA attached with the desired gene. So if desired gene is absent within a cell given, then it will be only transformant. Otherwise, if it contains the target gene, then it is called recombinant. And here the non-transformants do not receive the plasmids. So here the non-transformant is the diagram C depicting the non-transformant. And here non-recombinant will be, the non-recombinant do not have a target gene, but it will have a plasmid. So here it is B is a non-recombinant. In the second part of this question, they have asked three definitions. The first one is transformant host. Transformant host is a host that has taken up a vector DNA from its surrounding. And the second definition they have asked for non-recombinants. Non-recombinants are the host which have not received a vector from its surroundings. And the third definition they have also asked for third definition, that is define recombinants. Recombinants are the host which have received a vector DNA with a desired gene, that is vector DNA and gene of interest. If they present together, then that is called recombinant DNA. The host which have received it is called a recombinant host. Remember, these definitions are not given directly in textbook, but the concept is there. So learn it properly. Thank you.